on diabetes using implantable metastatic niche, and I will get it started by sharing the screen. Just give me a moment. Can you all see my screen?
Yes, it's visible.
Okay. Is it in full screen mode now?
Yes, yes, yes.
Okay, fantastic. So I'll be talking about my project, which involves developing implantable niche to detect metabolic changes during the onset of type 1 diabetes. So before going into what I am doing on the biomaterial side of things, I'll just like to give a brief introduction about what type 1 diabetes is, the pathogenesis, and burden related to it. So type 1 diabetes, unlike the type 2, is an autoimmune disease which has been linked to various genetic mutations. And what happens in this type 1 diabetes is the immune cells of your body, which in normal circumstances is supposed to fight off bacteria or infections or viruses, they undergo some rewiring, which causes them to go and destroy the pancreatic beta cells. And as a result of destruction of the pancreatic beta cells, the glucose, the insulin level in the body comes down, and as a result, the glucose level shorts up. So this is the pathogenesis, what happens in type 1 diabetes. And currently, there are more than 9 million people all over the globe who are suffering from type 1 diabetes, and the incidence rate is supposed to go all the way up to 15 million by the year 2040. So that's why it's so important to detect this disease early enough and to cure it. Just before I go ahead, I would like to talk about what is the specific age group which is being affected by type 1 diabetes. So naturally, in human beings, the primary age group is children of the age of 3 to 9 years. That's where they naturally get detected with type 1 diabetes, or sometimes it often happens during the teen and the adolescent years also. So what the problem is with the current clinical practices, which involves having a blood-based autoantibody test for the detection of type 1 diabetes, there are several caveats, and this is what I would like to spend a moment on before going ahead. So suppose, for example, there is a group of type 1 diabetic susceptible individuals as shown over here on the very left. Now, this can be regrouped into further three kinds of groups. One, where they show no appearance of autoantibodies and they are normoglycemic throughout their life. So it means that you have the genetic mutation in you, which makes you susceptible or at risk for type 1 diabetes, but you neither develop the autoantibodies in your blood nor develop any symptoms. So you're healthy throughout your life. You just have the mutation. This is the first category of group. The second category of group is over time they develop these autoantibodies in the blood, they get detected with the autoantibodies, and also over time they progresses to become type 1 diabetic. So this is the group which suffers from the disease pathological burden. The third case is what becomes challenging to identify, is that this particular group, they develop autoantibodies in the blood, but they do not progress to type 1 diabetes and they remain normoglycemic throughout. So it's like you have the mutation, you develop those autoantibodies in the blood, but you do not progress to the final disease states. And that's why it's a bit challenging because with the current blood-based autoantibodies, it's difficult to identify simply on basis of those autoantibodies that which of them will progress to diabetic versus which of them will remain in the normoglycemic states. That's why it's important to develop a biomarker panel which is more specific to the disease onset and not just gives you a predictor of risk, so as to speak. So that's why what I was like saying over here, important to identify mechanistic biomarkers very much specific for the onset of type 1 diabetes, because this current clinical based test, which are based on these blood based autoantibodies, they tell you, okay, you have those autoantibodies in the blood, there is a risk that you're going to become diabetic, but there is no direct confirmation that yes, you're going to progress to type 1 diabetes. And unfortunately, all the therapeutics which have been prescribed for type 1 diabetes, unless and until you develop some symptoms like hyperglycemia, you won't be treated with those because they have some other side effects because they are kind of immunosuppressants. That's why it's important to identify the exact specific cohort or group which is going to progress, so that they can be given the early treatment. Because if you give the same treatment to people who are going to remain normoglycemic, they may have some other side effects from the treatment. So characterizing the transition into type 1 diabetes, needing for understanding the mechanisms. So there are basically four kinds of factors which plays a key role in the type 1 diabetes incidence. First is the genetic predisposition. And just to make this thing clear, unless and until you have the genetic mutation in the genetic mutation in you, you won't progress to type 1 diabetes. So that is the first, like the checkpoint. So all people who progresses to type 1 diabetes, they must have the genetic mutation. Like that's the minimum criteria. The second thing is, even if you have the genetic mutation, always will not progress to type 1 diabetes. Some of them remain normoglycemic. So what are the other factors which are playing role in this progression to type 1 diabetes versus remaining in the healthy state? So those can be classified into the broad umbrella of three categories. One is the immune cell rewiring in the pancreas microenvironment, how the immune cells are changing their phenotype and proceeding towards more inflammation state or anti-inflammation state. Their metabolism has also been shown to provide a significant effect on the progression versus non-progression. And the final thing, which I won't be covering in my topic today, but is a very important thing for the on type 1 diabetes incidence, is the external factors and the gut microbiome, where it has been shown that the microbes in the gut also interact with the immune cells in the pancreas, and they play a significant role in the onset of disease. But primarily, I will be talking today about the immune cells rewiring in the pancreas, and also slightly touching on the metabolism aspect, because that's what my work covers. So monitoring the immunometabolic changes can help in predicting the T1D onset. That was the thought process. And to overcome this, this is the two main aim of my work currently. Firstly, to define the stages of type 1 diabetes on basis of the immunometabolic changes. For example, like type 1 diabetes has various stages: stage 1, stage 2A, stage 2B, stage 3. Currently, all these stages are defined by the extra, like the phenotype which is the person shows. For example, in stage 1, they only have the genetic mutation, no symptoms at all. Stage 2, they show those autoantibodies and maybe slightly dysregulated blood glucose level. And stage 3 is where they show hyperglycemia, the blood glucose level has shot up and the person is actually suffering from symptoms. But it's also important to actually go ahead and define what is exactly the biological changes which are happening in the pancreas at these three stages. And that's what I'm currently focusing on. And to define this, we are using an implantable sensor in T1D susceptible individuals to identify early changes for therapeutic intervention. So the tool which the Shell app uses for monitoring the T1D progression is scaffolds. So before going ahead, I would like to talk a little bit about scaffold and its comparison with blood-based tests, that why we're using a scaffold-based sensor as compared to simple blood draw-based tests. So the what should I say? The advantage of using scaffolds over a normal blood-based test is scaffold helps to capture the dynamic immune changes over time as compared to blood, which gives you basically kind of a snapshot at that particular moment when you're drawing the blood. So how we engineered the scaffold is as follows. The scaffolds are made up of PCL and they have a microporous structure. So because they have this porous structure, we implant them right underneath the skin. And once they're implanted, it takes around a week for it to undergo vascularization. So basically the blood vessels start growing inside the scaffold as shown over here. And once this blood vessel has grown in and the vascularization has taken place, then what happens, which is very interesting, is that it undergoes, what should I say, infiltration by various kinds of immune cells, like primarily by the innate immune cells like macrophages, DCs, neutrophils, and all, but also by T cells and NK cells and so on. So as compared to blood, which we are drawing at that particular time point, with scaffold, what we do is, suppose in the mouse model, we put it underneath the skin of the mice, we wait for around 2 to 3 weeks, and then we explant the scaffold and put a new one, and then basically we do the bulk RNA sequencing and single cell RNA sequencing. Now you can really understand that once the scaffold is inside the skin of the mouse or inside the body of the mouse for 3 weeks, it captures all the immune changes which were happening inside the mouse for this span of 3 weeks, as compared to blood, which is more of a snapshot of the change, as I mentioned. And also similar to the blood, this is kind of very non-invasive because you just put it right underneath the skin. There is no like need to go and take the biopsy of the pancreas, which is a very sensitive organ. So in that way also, it's a lot less non-invasive as compared to many other tests. So what we did over here is as follows. We took a model which is the non-obese diabetic mouse. So why we took this model is as follows. So the female non-obese diabetic mouse, they have the genetic mutation in their genes which make them susceptible to type 1 diabetes, and this is the model which mimics the human pathogenesis the closest. So this mouse have the mutations in their genes, and what happens is over time, for example, suppose I start with 100 mice, just example, 80 of them will become diabetic, 70 to 80 of them, and 30 of them won't become type 1 diabetic. So this is exactly what happens in human beings also, that where suppose in a population of 1,000 people where all of them have the genetic mutations, we see 700 to 800 becoming diabetic, whereas 200 to 300 remaining in the normal glycemic state throughout their life. And that's the reason we took this NOD mouse model, which because it mimics the human conditions so closely. And what we did as is follows: we implanted the scaffolds underneath the skin of this mouse. We started with a cohort of 20 mouse, and we explanted them at 5 time points, starting from T0, which was around 6 weeks of age, T1, T2, T3, T4. T4 is the final stage where like some of the mouse have become diabetic, some of the mouse has not become diabetic. And as you can see, T1, T2, T3 are 3 weeks before diabetics, 5 weeks before diabetic, and 7 weeks before diabetic. So now we have explanted scaffold from this mouse at various time points, at these 5 time points. And at the end of the study, we had one group which had progressed to diabetic, the group which has not progressed to diabetic. Now, before going ahead with the results of what we did with the scaffolds, I will just like to point out why this study was so novel is because currently the testing of type 1 diabetes is what is the most of the people do is that at all these time points T0, T1, they sacrifice the mouse and take the pancreas out and do the sequencing, histology, and all of the stuff. But what happens is once you're sacrificing a mouse at early time point, you actually do not know whether it was going to progress into type 1 diabetic or it was going to remain in the healthy state throughout. But what happens with this scaffold based approach is you can monitor a mouse throughout the entire time span without the need to sacrifice it. And at the end, you have both the groups as well as data from all the longitudinal time points. So this is the novelty of the study. Basically, we're monitoring the same mouse over the entire time span. And we had started with a cohort of 20 mouse. I think on the top of my head, I think 12 of them became diabetic and 8 of them did not become diabetic. So we got an incidence rate of around 60%. So then what we did was from all these 5 time points and the two groups, the diabetic and the never sick NOD mouse, we took the scaffolds and did a bulk RNA sequencing of the scaffolds in the hope to get genetic markers based of scaffolds which can predict the onset of diabetes early enough. And this work on the experimental side of things was primarily be done by Jessica King, who is a senior graduate student in our lab. And then what we did was more on the computational side of things, where we identified a signature of diabetic versus never sick mouse using an elastic net-based machine learning model. So this we developed this model using the longitudinal data from both the diabetic mouse and the never sick mouse for the 5 time points starting from T0 all the way up to T4. And using elastic net, we wanted a panel of biomarker genes, around 20 or 30 biomarker genes, which can predict the onset of diabetes as early as 6 weeks of ages. And what we saw was that we got a panel of around 32 genes, which we are currently narrowing down. And this group was not only able to identify the diabetic cohort shown on the left from the never sick cohort shown on the right in yellow, but it was also able to identify which of them were going to become diabetic early as compared to the late diabetic stages. So it was not only about predicting which is going to become diabetic versus which is not going to become diabetic, but also giving you an idea of which is going to becoming diabetic early enough as compared to the ones which are going to be the late diabetic stages. Because just to clarify, the mouse has different like onsets of type 1 diabetes. Some of them become diabetic as early as 12 to 13. Some of them take all the way up to 18, 19. Similar cases in humans also, because some of them may become diabetic at the age of 3, 4, whereas some may develop diabetes in teenagers as late as 18, 19. So this is where we currently are, and this proves that scaffold can act as sensors for predicting the onset of T1D. And this was followed by some GSC analysis both on the cell type side of things and the metabolic pathway side of things, where we wanted to see that what are the cell types which are changing over time in this scaffolds based on these gene signatures, and also what are the metabolic pathways which were showing changes over time. So we took this bulk RNA sequencing data from the mouse which is progressing to diabetes versus the mouse which is not progressing to diabetes and remaining healthy throughout. And at each time point, T1 to T2, we compared the progressor versus the non-progressor. So if it is red, it means it is upregulated in the progressing. If it is blue, it means it is downregulated in the ones which are progressing to type 1 diabetes. The size of the bubble basically denotes the log p value. So if it is bigger, it means it's more significant as compared to the smaller bubbles. So what we saw was as follows. On the cell type side of things, macrophages, T cells, and dendritic cells were some of the cell types on basis of which were changing on basis of the gene signature which we saw. And this is also relevant for us because on basis of the literature and the changes happening in pancreas during the onset of type 1 diabetes, which I will talking about in a moment, macrophages, which are innate immune cell type, and T cells and DCs, which are more on the adaptive side of things, they play a critical role in the onset of type 1 diabetes. And these cell types were also showing up from the gene markers in our scaffolds, which was nice to see. The second thing was on the metabolic side of metabolic pathway side of things, where we saw that pathways related to fatty acid metabolism, TCA cycle, and amino acid metabolisms were some of the pathways which were showing changes between the healthy versus the diabetic group over time. And this was interesting because when we looked at the literature, all these pathways which were changing were also related to the immune cells rewiring. So currently we think that these pathways which are changing is because these immune cells are themselves undergoing rewiring during the onset of type 1 diabetes. And when they undergo rewiring, they change their metabolic pathways from OXPHOS to TCA glycolysis. And that's the result of which this dysregulated pathways are showing up during the onset of type 1 diabetes. So if I put it in a single word, so this shows that scaffolds indicate the integrate interplay between the immunological changes shown by the cell types over here and the metabolic changes shown by the metabolic pathway changes over here. And before now, what we are currently doing is we have identified, okay, the scaffolds are able to predict changes in diabetic versus non-diabetic mouse, and also has given us a biomarker panel which can predict early enough whether the mouse is going to become diabetic or it's going to remain healthy throughout. But what we want to see is more on the biology side of things, that okay, what is exact immune changes which are happening in the pancreas which causes some mice to become diabetic versus some mice to remain in the healthy state. And that's to answer that question, we will be currently conducting a study. But before I talk about that study, I will be talking about a very similar study which was done in the past, but it has some limitations which I'll be talking about later. But first, let me go ahead and describe this study. So this study was done by Jakarov et al, and this study was published in JEM, Journal of Experimental Medicine, in the year 2020. So what they did over here is as follows: that at three time points, 4 weeks, 8 weeks, and 15 weeks, which is the initiation. They have termed it as initiation, amplification, and destruction. What does initiation means? They say that the onset of type 1 diabetes or the initiation of the immune cell types into the pancreas starts around 4 to 6 weeks. That's why they have termed this stage as initiation stage. This is amplification, 8 weeks. That's where they start amplifying, and destruction around 15 weeks is where they undergo full-fledged destruction. So at each stages, they pooled mice. For example, at each stages, they took around 8 to 9 mice, extracted their pancreas, and pooled them together. Then what they did was a fact-based isolation. So what does it mean? Basically, on basis of using fluorophores and flow cytometry, they separated all the non-endocrine cells and only took the endocrine cells and the leukocytes, basically the immune cells and the endocrine cells. That's what they took, and they used those cells for single cell RNA sequencing. So once the single cell RNA sequencing was done, this is what the results came out to be. At around 4 weeks of age, this pink one is the endocrine cells, which is not that important from the pathological perspective. But what is important from our standpoint is the immune cells, like the MAC cells, T cells, mesenchymal cells, CDCs. So all the way from here. So what we see over here is as follows. As we move from 4, 8, and 15, the T cells shown by this kind of green, not dark green exactly, like this particular color. As you can see, it increases over time, whereas the macrophages, which were shown over in the yellow ochre kind of shade, this is more in population in the early phases compared to 8 weeks, where it slightly decreases, and around 15 weeks it undergoes further shrinkage. So this is exactly what we also expected. Around 4 weeks, the macrophages are the ones which are the dominant cell type, whereas 8 weeks and 15 weeks, T cells are the ones which is becoming dominant cell types. So the macrophages is kind of decreasing over time, and the T cells are kind of increasing over time. And this makes sense because before going ahead, let me just take a moment to explain that why this is logically makes sense is because in the early stages of type 1 diabetes during the initiation period, the macrophages are the ones which helps in the signaling mechanism. And basically once they identify the beta cells, proteins or autoantibodies, they help to basically call the T cells into the pancreatic microenvironment. And once the T cells come into the pancreatic microenvironment, they interact with these macrophages. The macrophages present the islet specific peptides to these T cells. And once the peptides are presented, the T cells become specific to the uh, beta cells in the pancreatic islets, and then the T cells are the basically the ones which go and kill the beta cells in the pancreas. So that's why they are increasing towards the end time point where the destruction of the pancreas is taking place full-fledged. Whereas towards the early time point, it is the macrophages which are higher in number because they are the ones which are playing the role in the initiation of the disease. So that what it makes sense. The DCs also are a very important cell types because they basically kind of play a role between the communication of the macrophages and the T cells, and that's why they become very important in this entire pathological onset of T1D. So now coming to the limitations of this particular study was that since they pooled mice at all these 3 time points, basically they took 5 to 8 mice or 8 to 9 mice, they extracted the pancreas and pooled all of them together. They were not able to distinguish between progressors and non-progressors. What do I mean by that is as follows. For example, when they're taking these mice at 8 weeks of age, or even for like 4 weeks, 8 weeks of age, or even 15 weeks of age, they are not yet showing high blood glucose level because they have no symptoms, because they're just like at risk kind of. So when you are like pooling all of them together, there will be some mice which was not going to progress to type 1 diabetes and they were going to remain healthy throughout, and some of them were going to progress. So it is kind of, you are what should I say, putting both the control group and the disease group together. But I understand like why they did is because currently on basis of the current blood markers, it's almost next to impossible to go ahead and distinguish whether one is going to become diabetic or not going to become diabetic early enough. And that's why it is important to go now that we have these scaffold-based biomarker panels, which can predict the onset of diabetes as early as 6 weeks. We want to use these biomarkers to see that, okay, we will not only will go and identify mice early enough that, okay, this mouse is going to become diabetic as we go along. This is not going to become diabetic as we go along. So extract the pancreas from both of them and do the single cell RNA sequencing on both of them separately, and then see a comparison of control versus the progressor to identify changes, immunological changes, which are very specific to the onset of T1D. And that's exactly we are currently doing. Is we are targeting 4 time points. One is the initiation around 6 weeks of age, which is the stage 1. The stage 2A, or the early stage 2A, which I call, is around the early pre-diabetic stage, which is around 8 to 9 weeks. And the late pre-diabetic, or the stage 2B, is around 11 to 13. And the diabetic time point is where the mouse actually shows symptoms versus the control mouse, which is still healthy. So what we're doing is we're using scaffold-based biomarkers to distinguish progressor versus the healthy or the non-progressor for the early stages. We are extracting their pancreas. We are doing this, isolating the immune cells, doing a FACS sorting, and then we are running a single cell RNA sequencing on them to identify the immunological changes in this mouse. So what we plan to do with this study, and this is currently in process, not yet done. Firstly, we want to see the trajectory analysis of macrophages and other immune cell differentiation using RNA velocity. So as I told you, the macrophages are the ones which play a very critical role in the initiation of type 1 diabetes. Now what happens is, macrophages, just to clarify this point, macrophages are always present in the pancreas of a mouse or even any human beings. So those macrophages are called islet resident macrophages. They are not the ones which are pathological. They are present even in normal mouse or normal human beings also, and they help to maintain the homeostasis. But what happens is, during this mice which becomes type 1 diabetes, this macrophages changes their immunological phenotype, or there are macrophages which are coming into the pancreas microenvironment from the blood or the lymph nodes, and they are the ones which are playing a role in the pathogenesis. So we want to see that when these macrophages, which are islet residents or which are coming inside the pancreas, and they are changing their phenotype from the naive state to this pathological state where they're causing inflammation, how are they changing, what are the intermediate subtypes they're undergoing through, and what are the basically the pathways which are helping them into undergoing this rewiring from the naive state to this pathological state. The second thing which we are doing is, or we are planning to do, basically we are trying to identify a immune cell based pro or anti-inflammation gene sets correlated with the disease progression. So as you know, type 1 diabetes is caused by inflammation by the immune cells. And that's why we're trying to identify gene sets. Gene set basically means kind of like not exactly pathways, but similar to pathways course. So we are trying to identify what are the pro and inflammation related pathways for the immune cells, which are causing progression to type 1 diabetes versus the ones which are remaining healthy. So this is what we plan to do in the what should I say, diabetic versus non-diabetic mice. We plan to use an algorithm called Spectra, which is a supervised based gene set analysis. It basically helps in deconvolution, and using the anti-inflammation and pro-inflammation phenotype, we want to see that whether the immune cell based pro and anti-inflammation gene sets can help in distinguishes T1D onset versus the healthy conditions. And the final thing is basically to identifying the metabolic flux rewiring contributing to the inflammatory phenotype, again in macrophages and also in T cells. So again, what I was telling you right just before is that this macrophages changes their phenotype from the naive state to this pathological disease state, which causes them to go and like basically call the T cells into the microenvironment and causes all the destruction of beta cells. So when they change their phenotype from the naive state to the disease inflammatory state, they undergo certain metabolic rewiring, and we have some idea that they change from oxphos to aerobic glycolysis and lactate pathways. So this is where again we will be using single cell RNA sequencing data, and we will be developing a what should I say, gene-based metabolic reconstruction model, and this will be doing a flux-based analysis using a software called Cobra in MATLAB. Basically, it's a constraint-based flux regulation analysis where you put the constraint and do the flux analysis by solving a bunch of ODEs for each mice. This will help us to identify what are the exact specific pathways fluxes which were changing over time. The previous pathway analysis, it's more general. Like for example, it will only tell you glycolysis pathway is upregulated or downregulated. The flux-based analysis will more pinpoint, okay, in glycolysis, it's the lactate to pyruvate step which is getting upregulated or downregulated. So it's more about going and identify which is the specific step in the pathway, or which is the specific enzyme which is getting dysregulated in the what should I say, rewiring of these macrophages or even other T cells, because then that helps us to identify, okay, this is the exact pathway mechanism which is helping in this transition from anti-inflammation to pro-inflammation in these macrophages or even other cell types. So this is kind of what we are currently doing. And before wrapping things up, I will just like to once again give you an idea of the why we are doing this, what is the big picture, and need for doing this is because currently out there in the clinic, the way they have defined stages of type 1 diabetes is as follows, where they have stage 1 is the early stages, stage 2A is the early pre-diabetic, where there is some sort of dysglycemia. Dysglycemia means the blood glucose levels are not normal. They are not too high, but they are not normal, just to put it in simple terms. But there is no clinical symptoms. The late pre-diabetic stage is also kind of similar. Dysglycemia and no clinical symptoms. And stage 3 is where already hyperglycemia has set in. 40 to 80% of the beta cell has been destroyed. So it becomes kind of a point of no return, fully speaking. And that's why it is very important to go ahead and define these changes like states early enough so that we can understand what are exactly the immunological changes which are happening in the stage 1, stage 2, stage 2B, on basis of which you can classify the progressor versus non-progressor, but also target those pathways and cell types so that we can go ahead and take some therapeutic measures. And this is exactly what we're doing. This is kind of the proposed mechanisms, and it can change as we get results. So we propose that during the early stages, the beta cells is stressed out, and that's why it causes the macrophages, the interaction of the beta cells with the macrophages cause them to change their phenotype from the naive state to kind of this pro-inflammation stages. In the early diabetic stage, the macrophages basically signals to the T cells to infiltrate into the pancreas, the kind of destruction of beta cells kind of starts. The late pre-diabetic stage, the beta cell destruction continues, and it's almost kind of the beta cell tries for one last time to kind of save it, but it's not able to do so in the ones which progress type 1 diabetes. And the diabetic time point is basically the one where destruction has already taken place. So this is kind of the big picture where we're trying to identify the specific immunological changes which are key drivers of onset of type 1 diabetes. And for doing this, we are using the scaffold-based biomaterial sensors to identify the progressor versus the non-progressor, and then going ahead into the pancreatic microenvironment to characterize these changes. So if I put it in one word, it's basically using a biomaterials-based approach to answer a more biology-specific question. Try to put my research in one term. And exactly what I was saying, mechanistic understanding of stage 1 and stage 2 can lead to earlier therapeutic intervention. Before I wrap up, I'll just give a snapshot of some of the other projects which we are doing. So one of the projects which is very interesting, at least to me, is using the scaffolds as sensor for islet transplant rejection. So this is basically the next stage. For example, somebody has already become type 1 diabetic. Then what do we do? Like there is no point, like the nanoparticles won't work, the anti-immunosuppressants won't work. So in that case, what you have to do is go ahead and do a transplantation. So basically you have to transplant the islets, and the islets then will help in generation of insulin and make the person remain in the normal state. But as you can understand, with any transplantation study, because as you have heard, I'm sure many of you have heard that when people undergo kidney transplantation, which is somewhat common, there is a high chance of rejection. Like kidney transplantation, high transplantation, there's a high chance of rejection because of the immune cells of your body perceiving the transplant as a foreign object, and as a result, it tries to reject it. The same thing happens with islets. Even after taking care of all the immunological HLA matching and all, there are obviously some differences, and that's why once you implant the islets into the mice, they give some immunosuppressants over a span of time. But in spite of implanting the islet, giving them the immunosuppressants, it always happens that, suppose for example, you have implanted the islets in around 10 mice, 6 of them accepts the islets and start functioning normally, but 4 of them rejects it. So over there, we're using the scaffolds to basically predict the ones which are going to reject versus the ones which are going to accept. Because once we're able to identify which are the ones which are going to reject, we can give them a higher dosage of immunosuppressants so that basically they do not reject. Other thing, on the cancer side of things, we are using this cancer and the scaffold as to identify what are the hallmarks of cancer, and this is done with collaboration with one of the grad students in our lab, Rebecca Pearls. Another of my study, which is was talking about the nanoparticle side of things. So this nanoparticle therapy is very common in autoimmune diseases, where once you give these nanoparticles, they basically help in tolerizing the T cells. What do I mean by tolerizing the T cells? It means that basically the T cells will no longer go and kill the beta cells, if I put it in simple terms. But exactly as it is the case in this islet transplantation, some mice respond to the nanoparticle therapy, some mice do not respond to the nanoparticle therapy. And we are trying to identify which are the mice which is going to respond, which is versus the ones which are not going to respond. And can we change the dosing of these nanoparticles to like make a better response in mice which are kind of resistant to the therapy. And the final thing, which I'm also working on with one of the postdocs in our lab, we are trying to develop a package in R. This is more on very computational side of things, where we are using this elastic net-based machine learning model for a multi-group biomarker analysis. So if you have more than 3-4 groups, this package is kind of suitable for those kind of data sets, and uses a machine learning algorithm where, suppose you start with around 10,000 or 40,000 genes for 5 groups, it will help you to identify which are the 20 or 30 genes which is helping to distinguish between all the 4 or 5 groups over time span. It takes into account all the pre-processing steps followed by pathoanalysis and everything, speaking. So this is currently we are working on. Hopefully in another 2-3 months this should come out. But yes, this is all from my side. And before wrapping up, I will like to acknowledge my current lab, Professor Lonnie, who is my PI, and also all the members of my lab who have been very helpful, my undergrads who work with me. And that's all. And the floor is open for question. Thank you.
I will ask the participants if they want to ask any question. Okay. Okay, so I think there is one question in the chat. So I'm saying, so can you please explain? So this person she wants to know that can you please explain the how scaffold can act as a biomarker? So
Okay. I think interesting question, and nice. Thanks for asking this. Just for me to clarify, I'm not I'm not saying that scaffold act as biomarkers. The scaffolds have immune cells and other kind of cell types in them. So we take the scaffolds out and take the cells of the scaffolds and then do a bulk RNA sequencing on the cells from the scaffolds. And using the genes from the cells, they are the ones which act as biomarkers. Scaffold is just what should I say, a synthetic environment which we are creating underneath the skins, which is helping in, what should I say, the accumulation of all these immune cell types. And once these immune cell types have accumulated over a span of 3-4 weeks, then we are taking out the scaffolds, extracting all the immune cells or the cell types from there, extracting the RNA from there, and then doing the RNA sequencing and developing the biomarkers from those. So it is the biomarkers that develop from the cell types residing in the scaffold, if that answers the questions.
so Jyotirma, I think she is now she means she has one addition to that question, so she's now asking that how can scaffold trap the cells? So you were just saying
Okay,
that how they're able to trap the cells.
so this is thanks for asking this. This is exactly what I was trying to emphasize in the early part of the presentation. I think like I could not drive the point clearly, but I'll just go over this once again. So what happens is these scaffolds have a microporous structure, and these scaffolds are made up of PCL, polycaprolactone, and they have a microporous structure. So once you implant the scaffold underneath the skin of the mice, they undergo vascularization. What do I mean by that? Blood vessels grow inside the scaffold and they become kind of integrated with the tissue. For example, when you're taking out the scaffolds, it's not like you can easily take them out. They become kind of integrated. So we have to like kind of cut them apart. So once they undergo this integration, then the immune cells infiltrate into the scaffold because they have now have a rich supply of oxygen and the blood vessels, creating an environment conducive for the immune cells or any other circulating cells, to speaking. And that's where they are able to create this microenvironment where it is reaching immune cells or any other kinds of cells, and that helps in detection of the disease. To clarify, just to make, I think I've not told this point. We first started with the scaffolds for detection of cancers, and our work has like our lab has done a lot of like scaffold related work for the detection of cancer. And now we are currently expanding the scaffolds for other disease model. I'm working on T1D, but we have also used scaffold for this transplantation of heart and also for what should I say allergies. So this is like kind of what we're doing.
Uh, the next question. So there is another question in the chat, which is that which material you are using to make the scaffold. So if you
Yes,
can
just
explain.
as I told you, it's polycaprolactone, so PCL. So basically, we start with a PCL salt dispersion. We first make the PCLs like the scaffold structure, which is around 5 millimeters in diameter. So it's very small. It's 5 millimeters in diameter, small, very thin width, around maybe less than a centimeter, truly speaking. Even less than that, I think zero point 0.5 or zero point 0.1, like 2-3 millimeters, not more than that. So once we press it at a very high pressure and temperature, and once it is pressed, it is kind of this circular disc structure. We leach it overnight or even more than that for the salts to get out. And once the salt is got out, it is a porous structure, and it is followed by the what should I say? Um, uh, the processes of cleaning it up and sterilizing process, and then once it is sterilized, we put it in the mouse. Any other questions?
So there is another question that factory which causes the change in the immunological phenotype in macrophages, is it known or is it not known?
It's not known. So I the factor which causes immunological change in macrophages, it's not known in T1 type 1 diabetes. Similar studies have been performed in cancer. So from the cancer perspective, we have more idea, and we are trying, like I'm sure, although we cannot do a perfect analogy between autoimmune disease and cancer because they're very different models, but there are some similarities we're trying to explore. But from an immunologic, like autoimmune disease perspective, it's not yet well defined.
So I also have personally had a question that you mentioned about the difference between the infective and the non-infective macrophages resident, right? Uh, the two types of macrophages. The one is the infective and the non-infective type.
You mean inflammatory and non-inflammatory, right?
Inflammatory and the non-inflammatory type. So what is the experimental way for you to differentiate between these two?
Oh, yes. Thanks for asking this. And like, I should have given a bit more background on that. So that's why we're using single cell RNA sequencing So what single cell RNA sequencing does is, for each cell, basically it goes inside each cell and does the gene profiling for each cell. So basically, suppose if you sequence 10,000 cells, for each cell you will have the expression of all 40,000 genes. So now this pro-inflammatory versus the non-pro-inflammatory macrophages, they have specific gene markers which defines their phenotype. For example, pro-inflammatory macrophages have INOS related macrophages, INOD related gene signature, whereas the non-pro-inflammatory ones have ARG1 plus related genes. So then we go ahead and look at which are the cell types which are expressing these pro-inflammatory related genes, and we classify them as the pro-inflammatory macrophages versus the ones which are showing, suppose, high ARG expression. We are classifying it as anti-inflammatory macrophages. So that's how the annotation is done, if that makes sense.
Thank you. So do anybody else have any more questions? So you can either speak out or you can write it in the chat box. So Jyotirmoy, I don't think anybody else have any more questions currently. So it was a wonderful session from you. And you have shown some of the, means you have shown the importance of the immunological cells in the diabetes progression, which is a very good thing. And also you have also shown some of the good, very good, wonderful experimental conditions that you just mentioned about what are the advantage and disadvantages of some of the experimental conditions, showing some of the examples also. So so do you want to, means any last comments from your side, if you
Uh,
want to give?
I'll say thank you for having me. It was really nice to once again talk on a IIT Bombay platform after a long time. It was really nice getting to share my research, and it's a very wonderful initiative which you have started, and really nice to see that such kind of platforms have been made where people can come and share the research, and hopefully can people can get inspired by the work and like do more good research in the future. Fantastic work.
Thanks a lot. Also, best of luck for your PhD program
Thank
and
you.
continue your research.
Absolutely. Thank you so much.
Yeah.
Okay.
Yeah.
Have a nice
So we're ending
day.
the session here.
Okay. Absolutely. Bye bye.